Welcome all for the today's class. So we continue with our recall and revise series. The exams are quite close now, and you should be very ready for the examination. So this recall and revise series will definitely help you for the final examination. Today we will continue with the genetics module. We have almost 35 to 40 questions, and these questions will form the basic theme of your examination, which are frequently asked. So it may not be the same question, but the question will be on the same line. Okay. So, doctor, can you solve these questions?
Yes, sir. The
Yeah.
following have X-linked pattern of inheritance, G6PD deficiency, Klinefelter syndrome, androgenital syndrome, hemophilia B, familial hypercholesterolemia. Is there? 1 is there, 4 is there. Six and control syndrome. 1 and 4 is 2.
So
Option
these are the options.
B
So
and C.
yeah. So there is a, if you say 1 and 4, then you have 1, 2, and 3.
And 3.
Okay.
Yes.
Then which is not there? There definitely.
Androgenital syndrome is also androgenital. Sir, option C.
Yes, it is an option C. Adrenogenital is not a X-linked. Okay, so okay, that is how you go. Even familial hypercholesterolemia is not a. So G6PD deficiency is definitely X-linked recessive disorder, predominantly affecting males. Klinefelter is not disease; it's a chromosomal aneuploidy. Okay, so not a inherited X-linked disorder. Then adrenal syndrome is also false. Congenital adrenal hyperplasia, most commonly due to 21 hydroxylase deficiency, is inherited as autosomal recessive. Okay, then hemophilia B, that is X-linked recessive disorder. Okay, that is factor 9 deficiency. Then familial hypercholesterolemia, that is also false. Classical familial hypercholesterolemia is autosomal dominant, usually due to mutations in LDL. Okay, so important X-linked recessive disorder: G6PD deficiency, hemophilia A, hemophilia B, Duchenne's muscular dystrophy, Becker's muscular dystrophy, and green color blindness. Even in the examinations, if you get any example, generally these are the diseases which they use. Okay, so have a look over this particular condition: G6PD deficiency, hemophilia A, B, Duchenne's muscular dystrophy, Becker's muscular dystrophy, and red green color lines. And Klinefelter syndrome, chromosomal abnormality, not a X-linked inheritance. Congenital adrenal hyperplasia, it is autosomal recessive,
Yes.
and familial hypercholesterolemia is autosomal dominant. Okay, so next question, doctor. Yeah.
Yes, sir. In the female, only the X chromosome of maternal origin is active. Only the X chromosome of maternal origin is active. The bare body is a sex chromatin. No, about 80% polymorphonuclear leukocytes have drumstick chromatin. An extra X chromosome is associated with 2 bare bodies. An extra X chromosomes is associated with below. An extra X chromosome is associated with below average intelligence. Option 4 is right. And option B is wrong. 2 is wrong. Bare body is a sex chromatin. Is an bare body. Option D, sir.
Option
D.
D.
The only X chromosome.
Option A. Okay, that means only X chromosome of maternal origin. We have seen no Leonization, and there is a random inactivation of the X chromosome. It is not like that. If you listen to the my session, it is not a particular X chromosome coming from female or male, or the mother or father will. It is a random inactivation. Okay, so only the X chromosome of maternal origin is active. This is a wrong sentence. Okay, so except this, all are then showing this false. The bar body is a sex chromatin. Let us go into the answer key. Okay, so number of bar bodies equal to number of X chromosomes minus 1. So normally with two XX chromosomes, there will be one bar body. Okay, with three, there will be two XXY, there will be one, and there will be no bar body with Turner. Okay, so X inactivation is a random, not specifically abnormal, maternal. Then drumstick chromatin is present in only a small percentage of neutrophils. Bar body is inactive X chromosome, that is sex chromatin. Okay, only the X chromosome of maternal origin is active, that is a false sentence. X chromosome inactivation, leonization is a random in embryonic cells. Either the maternal or paternal X chromosome may remain active. The bar body is a sex chromatin, that is true. A bar body represents condensed inactive X chromosome material and is also known as sex chromatin. Okay, an extra chromosome is associated with two bar bodies, that is also true. Okay, so an extra chromosome shown as an extra bar body as there are two inactive X chromosomes. Okay, so in 47XXX, number of bar bodies will be equal to number of X chromosome minus 1, that is 3 minus 1 will be 2 bar bodies. Okay, and extra chromosome is associated with below average intelligence, that is also true. Okay, next.
In DNA,
This,
a codon is a sequence of 3 bases. All codons have an identified function. There is a greater variety of amino acids than there are different codons. There are greater variety of amino acids than there are different codons. The replication can be initiated at several different points along a chromosome. Replication can be initiated at several along a chromosome. Complementary pairing precedes messenger RNA synthesis. I'm going to say yes. The codon is a 3 base, true. All codons have identified function. There is a greater variety of amino acids and yes. Sir, option B.
option A is correct. A codon is a sequence of 3 bases, that is true. The genetic code is read in triplet of nucleotides codons, each specifying an amino acid or translational signal. All codons have an identified function, that is also true. There are 64 codons and all are assigned a recognized role. Amino acid coding codons, start codons is AUG, stop codons are UAA, UAG, UGA. There is a greater variety of amino acids than there are different codons, that is false. Okay, there are 64 codons but only 20 standard amino acids, so the genetic code is therefore degenerate. Okay, replication can be initiated at several different points along a chromosome, that is true. In eukaryotic chromosomes, DNA replication begins at multiple origins of the replication. Complementary pairing precedes messenger RNA, that is also true. So key numbers, 4 nucleotide bases, 64 codons, that is 4 cube, 61 sense codon, 3 stop codons. Okay, and 20 standard amino acids. So from examination point of view, more codons than amino acids. Multiple codons can code for the same amino acid, and eukaryotic DNA has multiple origins of replication. Next.
The following genetic disorders are inherited as autosomal recessive. Duchenne muscular dystrophy, Huntington's chorea, Tay-Sachs disease, retinoblastoma, achondroplasia.
Which is there, which is not there. At least if you know 3, then you should be able to guess it correctly.
There, Huntington's is autosomal dominant. So 1 and 2 is not there. Tay-Sachs disease, autosomal
DMD, is it a
X-linked recessive? No, it is not.
okay.
1 and 2 is negative. So we have A, D, and E. Retinoblastoma, autosomal achondroplasia. Tay-Sachs disease is autosomal dominant. The second, sir, Tay-Sachs disease. This is also autosomal recessive. Tay-Sachs disease is autosomal recessive. Okay. So 3 is positive in A and D. A and D. Then we have retinoblastoma and achondroplasia. Achondroplasia is, I think, so autosomal dominant. It has to be negative. Retinoblastoma. Fusion is not there. Autosomal Huntington is not there. Tay-Sachs disease is autosomal recessive. Okay. But I know achondroplasia is not there. Negative. And option D, sir.
Option
H.
A. DMD is false. DMD is X-linked recessive. Huntington's is also false. Huntington's disease is autosomal dominant. Okay, caused by CAG trinucleotide repeat expansion. Tay-Sachs disease is true. So Tay-Sachs disease is an autosomal recessive lysosomal storage disorder caused by deficiency of hexosaminidase A. Then retinoblastoma is also false, and achondroplasia is also false. Both are autosomal dominant. Okay. Then
Yes.
common autosomal recessive, Tay-Sachs disease, cystic fibrosis, phenylketonuria, sickle cell disease, beta thalassemia, congenital adrenal hyperplasia. So these are common autosomal recessive disorders. Common autosomal dominant, Huntington's disease, achondroplasia, Marfan syndrome, familial hypercholesterolemia, and retinoblastoma, familial form of retinoblastoma. And X-linked recessive, DMD, hemophilia A, B, and G6PD deficiency. Okay,
Hmm. The
next.
following are inherited as autosomal recessive condition: tuberous sclerosis, phenylketonuria, achondroplasia, sickle cell anemia, von Gerke's disease. 2 and 4 is there. 2 and 4 is there. So we have ABC. Achondroplasia is not there. So we have A and B. Tuberous sclerosis and von Gerke's disease. Gerke's disease. Von Gerke's disease is autosomal recessive. No? A and B. Sir, option B.
It should be A. It should
A.
be A. Because tuberous sclerosis is not tuberous sclerosis is autosomal dominant disorder involving mutations in TSC1 or TSC2. Phenylketonuria, yes. Okay, phenylketonuria is a classical autosomal recessive disorder caused by deficiency of valine alanine hydroxylase. Achondroplasia is also false. It is an autosomal dominant mutation in the FGFR3 gene. Sickle cell anemia is true. Sickle cell anemia inherited autosomal recessive disorder due to mutation in the beta globin chain. Okay, and von Gerk's disease is also true. Glycogen storage disease type 1. Von Gerk's disease due to glucose 6 phosphatase deficiency, autosomal recessive condition. So these are the autosomal dominant: tuberous sclerosis, achondroplasia, Huntington's disease, Marfan syndrome, familial hypercholesterolemia. Autosomal recessive: phenylketonuria, sickle cell disease, von Gerk's disease, Tay-Sachs disease, cystic fibrosis, congenital adrenal hyperplasia. Okay,
Hmm. Genes on
next.
sex chromosomes are responsible for the inheritance of G6PD deficiency, defective color vision, hairy ear rim, homocystinuria, Hurler syndrome.
So here they are asking for sex chromosome. Genes on the sex chromosome is for inheritance of. That means X-linked. They are asking. Okay.
2, 3, G6PD, 2, 3, homocystinuria. The option D.
What is G6PD deficiency?
X-linked recessive, defective alpha, X-linked recessive.
G6PD deficiency, X-linked recessive or autosomal recessive? Option A.
X-linked
Uh,
recessive.
G6PD deficiency, classic X-linked recessive, defective color vision. Sorry. Red green color blindness is typically X-linked recessive. Here, hairy ear rims, that is also true. Okay, Y-linked inheritance. It's not only X they are asking, sex-linked they are asking. So X as well as Y we should know. Okay, so hypertrichosis of the pinna, hairy ear rims, is a classic example of Y-linked Hollandric inheritance and therefore involves a sex chromosome. Okay, homocystinuria is false. It is going to be autosomal recessive because it will be some enzyme deficiency, which are mainly autosomal recessive. Okay, Hurler syndrome, MPS1. Okay, L-iruronidase deficiency, that is also autosomal recessive. Generally, enzyme deficiencies are all autosomal recessives. Okay, so X-linked disorders, G6PD deficiency, red-green color blindness, hemophilia A, B, DMD. Y-linked are hairy ear rims. And autosomal recessive, homocystinuria, Hurler syndrome, Tay-Sachs disease, phenylketonuria, etc. The question asked about gene on X sex chromosome, which includes both X-linked and Y-linked genes. Therefore, hairy ear rims should be marked true. Okay, you got it?
Yes, sir.
Next.
The following genes and chromosomes are correctly paired. HLA chromosome 6, clotting factor 8, 10 chromosome. G6PD dehydrogens, X chromosome, testis determining factor, X chromosome, XG blood group, X chromosome 1. G6PD is X chromosome, it is true. Okay, fourth one is wrong. We have ABC. HLA 6 chromosome. Chromosome 6. Clotting factor is also 8, is 10, 9 and 10. So 8. Figure 8. Also. ABC, no?
Not only solving the questions, but simultaneously concentrate on the how the questions are framed. What are what they are asking? Okay. So here they are asking gene and the chromosome combination with respect to disease. Okay.
Third one is X chromosome. It is true. Testis determining, it is false. Fourth one is wrong. Fifth one is also wrong. We have A and C.
Sir, B.
Clotting factor 8.
Clotting factor 8 and 10 chromosome. No, it is wrong. Option C, sir.
Option A, sir.
Monica says, Dr. Monica is saying option A, and Dr. Sansi option C. Okay, so correct answer is A. Okay, HLA chromosome 6, that is true. HLA complex, major histocompatibility complex, is located on the short arm of chromosome 6, 6p21. Clotting factor 8, X chromosome, that is true. The F8 gene is located on the X chromosome. XQ28 mutations cause hemophilia A. G6PD, that is glucose 6-phosphate deficiency, dehydrogenase, X chromosome, true. G6PD gene is located on the X chromosome XQ28 and is inherited as the X-linked recessive. Testis determining factor X chromosome, that is false. The TDF is encoded by SRY gene and is located on the Y chromosome, not on the X chromosome. XG blood group chromosome 1, that is false. XG blood group gene is located on the X chromosome, not on chromosome 1. Okay. So this HLA chromosome 6, factor 8 associated with X, G6PD, X, SRY, Y gene, XG blood group, X. Okay, so SRY or testis determining factor is on the Y chromosome. XG blood group is X linked, and both factor 8 and G6PD are located on the X chromosome. Okay. Next.
Yes, sir. In human, a haploid number of chromosomes is found in the RBCs, blastocysts, primary oocyte, first polar body, spermatozoa. It is found in first polar body. It is found in spermatocyte.
Sir, option C.
Fourth and fifth is correct.
And what about third?
Primary oocyte
No.
also.
It has diploid, no? Primary oocyte.
Diploid.
Primary oocyte has diploid, so it is wrong. Blastocysts, it is also diploid. It is also wrong. RBCs. It is
RBCs don't have nucleus.
Nucleus. Option A.
Option A, RBCs is a false. Mature human RBCs are anucleate and contain no chromosome. Blastocysts is a false. Blastocyst cells are derived from zygote by mitosis and are therefore diploid, 46X chromosome. Primary oocytes is also false. Primary oocytes are diploid and are arrested in the prophase 1 of meiosis. Although DNA has replicated, the chromosome number remains diploid. The first polar body is true. The first polar body is formed after the completion of the meiosis 1 and contains a haploid number of chromosome, that is 21. Spermatozoa, that is also true. Mature spermatozoa are haploid gametes containing 23 chromosomes. So haploid cells are spermatozoa, secondary oocyte, ovum, first polar body, second polar body. Many times this question has been asked. Okay, diploid cell, zygote, morula, blastocyst, primary oocyte, and somatic cells. Okay, a primary oocyte is diploid, whereas a secondary oocyte is a haploid. Okay. Sorry.
Yes, sir.
Just a-
Achondroplasia is yes.
huh.
Achondroplasia is a dominant trait. Babies with Down syndrome usually have 46 chromosomes. Congenital pyloric stenosis is commoner in babies affected parents than in the general population. All daughters of female carrier of red-green color blindness are themselves carrier. Hemophilia occurs in all the sons of an affected father. Achondroplasia is a dominant trait. True?
Hmm.
46 chromosomes. Trisomy, Down syndrome. No, it is not. It is not. Congenital two or three female carriers of red-green color blindness.
Sir, answer D. Sorry, E.
Okay.
Achondroplasia is autosomal dominant.
Hmm.
Babies
Yes.
with Down syndrome has 47 chromosomes.
So that is false. Okay. Then CHPS is common in babies of parents, affected parents, and all daughters of
Yes.
female carrier. So, Dr. Monica,
Red green
as
is
Option E only, sir. A.
they have given A, let us see. Achondroplasia dominant, correct. Babies with Down syndrome are usually have 46 chromosome, that is false. There are 47. CHPS is commoner in babies, that is true. Okay, all daughters of female carrier of red green color blindness are themselves carriers, that is false. So female XX passes the mutant gene to only 50% of her daughters, not to all daughters. Okay.
Yes,
Hemophilia
yes.
occurs in all the sons of an affected father. False. An affected father passes his chromosome to all sons and his affected X chromosome only to daughters. So that is false. So therefore, none of the sons, his sons, will inherit hemophilia directly via him. So this answer A is correct, and hemophilia occurs in all the sons of an affected father. Okay, so this is definitely wrong, no?
Wrong.
Okay,
Yes.
yeah. So again, this list we have seen. Okay, so pedigree traps you should remember. Affected father, all daughters will receive his X chromosome. Okay, then affected father, no sons will inherit his X-linked disorder. Okay, with respect to X-linked we are talking about. If the father is affected, all daughters will receive. If the father is affected, none of his son will receive. Okay, then carrier mother, 50% risk to the son and 50% carrier risk to the daughters. Okay, daughters will be carrier in 50% of cases and son will be at risk of 50% having the disease. Next.
Steps involved in identification of restriction fragment length polymorphism includes Western blotting or restriction enzyme digestion, Southern blotting, agarose gel electrophoresis, thin layer chromatography. Restriction enzyme. Sir, option E.
Option A. Western blot is false. Western blotting is used for protein detection, not for identification of DNA restriction fragment length polymorphisms. Remember, Western blotting is specifically for protein detection. Many times a question on Western blotting is asked. Then restriction enzyme digestion, true. DNA is cut with specific restriction endonucleases, generating fragments of various lengths depending upon the DNA sequence. Southern blotting, yes. After electrophoresis, DNA fragments are transferred to a membrane by southern blotting and then hybridized with a labeled probe. Agar gel electrophoresis, that is also true. Restriction fragments are separated according to size using agar gel electrophoresis. Then thin layer chromatography, that is false. Thin layer chromatography is used for separation of chemical compounds and has no role in standard RFLP analysis. Okay, so classical RFLP technique will involve DNA extraction, restriction enzyme digestion, agar gel electrophoresis, southern blot transfer, hybridization with labeled DNA probe, and detection of fragment pattern. Okay. So this you should remember: southern blot DNA detection, northern blot RNA detection, western blot for the protein. Okay, so this also is a separate SBA. Okay, so western blot is for protein, southern blot is for DNA. Okay. In genetics, you need to read the question very carefully. It may these questions may take little longer time for the solving. Okay, go ahead.
Yes, sir. DNA contains no cytosine, has a
Cytosine.
backbone, cytosine, sorry, has a backbone of ribose, is usually single-stranded in mammalian cells, is cleaved by restriction enzymes, is irreversibly damaged in vitro by heating to 75 degrees centigrade. Those three are wrong, sir. Those three are wrong. Is cleaved by, yes, it is cleaved. Is irreversibly damaged in vitro by heating to 75 degrees centigrade. True. It's true. Option D or A. Or A, option A.
Even I am
Option A.
confused between D and A. Sometimes, not all keys are correct. Okay, so we need to be very careful. Contains no cytosine, that is false. Okay, DNA contains adenine, guanine, cytosine, thymine. Okay, so that is definitely correct. Has a backbone of ribose, that is false. It contains deoxyribose. Okay, is usually single stranded, that is also false. Okay, DNA is double stranded. Is cleaved by restriction endonucleases enzymes, that is true, and is irreversibly damaged in vitro by heating to 75 degrees centigrade. Heating DNA to around 75 to 95 degrees causes denaturation, strand separation, but this is usually reversible upon cooling, renaturation or annealing. If conditions are appropriate, PCR routinely exploits this property. So there is no irreversible damage. Okay, so there is a reversible damage. Okay, at 75 degrees centigrade. So in DNA, sugar is deoxyribose. Bases are adenine, guanine, cytosine, thymine. Okay, they are usually double stranded DNA and function is mainly genetic storage. While RNA has got a ribose sugar, it is adenine, guanine, cytosine, and uracil. Okay, RNA has got uracil, usually single stranded, and the function is a gene expression. So from examination point of view, DNA contains cytosine, DNA contains deoxyribose, not ribose. Heating causes denaturation, not necessarily irreversible destruction. Restriction enzymes cut DNA at specific recognition sequences. Next.
Yes, sir. Mitochondrial DNA is located in the nucleus. Inheritance is patrilineal, is present in 2 copies per cell. Mutation causes cystic fibrosis, is involved in the control of oxidative phosphorylation. First one is wrong, second one is wrong, third one is wrong, fourth one is wrong. Is involved in the control of oxidative phosphorylation is true. So option A.
Option A is true, is located in the nucleus, that is false. Mitochondrial DNA or mDNA is located within the mitochondria, not in the nucleus. Inheritance is a patrilineal, false. Mitochondrial DNA is inherited almost exclusively from the mother. So maternal inheritance, because paternal mitochondria are not normally transmitted to the embryo. Is present in 2 copies per cell, false. The nuclear DNA is present as 2 copies, that is diploid in most somatic cells. Mitochondrial DNA exists in multiple copies per mitochondria, with many mitochondria per cell, resulting in hundreds to thousands of copies per cell. Mutation causes cystic fibrosis, that is false. Cystic fibrosis results from mutation in the CFTR gene on chromosome 7 and is an autosomal recessive disorder, not mitochondrial disease. Is involved in control of oxidative phosphorylation, that is true. Mitochondrial DNA encodes several proteins essential for the respiratory chain and oxidative phosphorylation. So mitochondrial DNA characteristics are circular double-stranded DNA located in the mitochondria, maternal inheritance, multiple copies per cell, encodes components of the electron transport chain. Mutations may cause mitochondrial myopathies, optic neuropathies, and other neuromuscular disorders. So from examination point of view, mitochondrial DNA is maternal, not paternal. Mitochondrial DNA is not located in the nucleus. Cystic fibrosis is due to nuclear gene mutation, CFTR. Mitochondrial DNA plays key role in oxidative phosphorylation. Okay, next.
Yes, sir. Concerning the genetic control of protein synthesis, mature mRNA contains introns. A codon has 3 base sequences. Each amino acid has a single codon. Transfer RNA has anticodon recognition sites. Each tRNA carries a specific amino acid. First one is wrong. Second one is
So
true.
answer C.
Third one is wrong. Fourth and fifth is correct. tRNA has anticodon. Option A, sir.
Let us see A or C. It is A. So base pairing rules: adenine, thymine, cytosine, and guanine for DNA. RNA, uracil, adenine, guanine, cytosine. Mature mRNA contains introns, that is false. In eukaryotes, introns are removed during RNA processing, that is splicing. Mature mRNA contains exons only. A codon has 3 base sequences, that is true. A codon is a sequence of 3 nucleotides that specifies an amino acid or start or stop signal. Each amino acid has a single codon, that is false. The genetic code is degenerate. Most amino acids are encoded by more than one codon, except methionine and tryptophan. Each have a single codon. Okay. Transfer RNA. tRNA has anticodon recognition sites, true. Okay. Transfer RNA contains an anticodon that base pairs with the complementary codon on the mRNA during translation. Okay. So each tRNA carries a specific amino acid, that is true. Okay, each tRNA is charged with a specific amino acid by a corresponding amino acyl tRNA synthesis. So protein synthesis, what does it involve? Formation of mRNA from DNA template first. Okay, so steps are: DNA double helix will unwound, then RNA polymerase binds to the promoter region, then complementary RNA nucleotides are added, RNA strand elongates, then transcription terminates, pre-mRNA undergoes processing, and mature mRNA exits the nucleus. Okay, so codon is a 3 base sequence mRNA. Anticodon is a complementary 3 base sequence on RNA, tRNA. Okay, mature mRNA has had introns removed. There are 61 sense codons for amino acids. 3 stop codons terminate translation. So mature mRNA does not contain introns. This is very important. Okay, most amino acids have multiple codons. Each tRNA carries one specific amino acid. So DNA template TACGGA CAT. mRNA, if this is the DNA template, mRNA will produce AUG, CCU, and GUA from this. Okay. So translation: methionine, proline, and valine. So with this template, you will get this codon from mRNA, and you will get this as a translation final product. Okay. So transcription occurs in the nucleus, catalyzed by the RNA polymerase. Introns removed during the processing. Mature mRNA contains only exons. Complementary base pairing is essential for mRNA synthesis. Remember, you need not know by heart the steps. You just understand few things, whatever you can understand, and then apply the logic from this. Okay. Next.
Yes, sir. When a man has hemophilia, 50% of his daughters will not expect to be carriers. 25% of his sons will be expected to be affected. Good medical control of his blood deficiency reduces the risk of good medical control of his blood deficiency reduces the risk of the condition in his children. His medical of his blood, no. His newborn child is likely to require an urgent blood transfusion. His sister has 50% probability of being a carrier. Your option C.
It is A answer. Okay. It is A. His sister has 50% probability of being a carrier. That is the only true. 50% daughters would not expect to be carriers. All daughters will be carrier. All daughters will be carrier. 25% of sons would be expected to be affected. This is also false. All sons will be affected. False. Correct. Good medical control of his blood deficiency reduces the risk of condition in his children. No relation with this. Okay, his newborn child is likely to require an urgent blood transfusion. Why spontaneous? There can be bleeding. There is no, generally these patients bleed when there is a trauma. Okay, so newborn child is likely to require urgent blood transfusion unless I don't know this fourth, but the fifth is definitely true. Okay, fourth we need to read about it. So 50% of his daughters would not expect to be carriers. That is false. An affected male XY passes his affected X chromosome to all daughters. So 100% daughters will be carriers, obligate carriers. Hence not 50%. 25% of son would be expected to be affected. This is false. Sons receive the Y chromosome from the father, not his X chromosome. Therefore, 0% of sons inherit hemophilia from an affected father. Okay. Then good medical control of his blood deficiency reduces. So son will be affected in 0% of cases. Daughters will be affected in 100% of cases. Good medical control of his blood deficiency reduces the risk of this condition in his children. Hemophilia is a genetic disorder. So treatment of the father does not inheritance alter the risk. No relation. His newborn child is likely to require urgent blood transfusion. Hemophilia is not associated with routine need for neonatal transfusion. Most newborns are asymptomatic unless bleeding occurs. Okay. His sister has a 50% probability of being carrier. Okay. If the mother is a carrier, obligate in many cases, each daughter has a 50% chance of being carrier. Okay. So this X-linked recessive affected male to daughter is 100%. Affected male to son is a 0%. Carrier female to son is 50%, and carrier female to daughter is a 50% carrier risk. Okay. Carrier female to son is a 50% affection. Carrier female to daughter is a 50% carrier. Okay, so these are the things which you should remember. Frequently ask. You will get these only permutation and combination. Okay, name of the disease may be changed, but overall the pattern will remain same. Affected father does not pass his disease to the son. Affected father makes all daughters carriers, and treatment does not change inheritance risk. Next, doctor.
Yes, sir. The following are X-linked disorders: myotonica dystrophica, myotonica dystrophy, Duchenne muscular dystrophy, hemophilia A, Huntington's disease, fragile X syndrome. 1, 2, 4 is not there.
So D, option D.
3 and 5th. Third one is X-linked fragile X. Option D, sir.
Option
D. Okay.
where just maybe a wrong. X-linked
Question.
myotonic dystrophy. I'm not sure. Myotonic dystrophy.
And,
DMD is
thing
definitely
recessive.
there. Yeah, myotonic dystrophy is a autosomal dominant. Okay, trinucleotide repeat disorder. Chromosome 19 is involved. DMD is true. It's a X-linked recessive. Hemophilia is true. X-linked recessive. Huntington's is false. It is autosomal dominant. Fragile X syndrome is X-linked XQ27-3. Okay, due to CCG repeat expansion in the FMR1 gene. So X-linked disorder list we have seen. Okay, fragile X is a X-linked. Then autosomal dominant myotonic dystrophy and Huntington's disease. Fragile X syndrome is X-linked but not classically recessive or dominant in simple Mendelian terms due to the dynamic mutation, but is still considered as a X-linked. Okay. Next, doctor.
Yes, sir. In the ovary, primordial germ cells are formed. Primary oocytes have completed the first meiotic division by birth. The majority of primary oocytes become atretic by puberty. Fewer than 10 follicles starts the process of antrum formation in each ovarian cycle. The second polar body is formed at ovulation. Option of primordial germ cells are formed. In the ovary, primordial germ cells are formed. No, it is formed in yolk sac. First is wrong. Primary oocytes have completed the first meiotic division by first meiotic division gets complete after ovulation. Option second is also wrong. The majority of primary oocytes become atretic by puberty. This is true. Fewer than 10 follicles starts the process of antrum formation. Now it is 10 to 20 actually. So it is also wrong. The second is also wrong. Option A, sir.
I think it should be fewer.
A or
No, fewer than no. It should be A.
A?
It should be A. Yeah. Primordial germ cells are formed. That is false. Primordial germ cells are formed in the yolk sac endoderm, not in the ovary, and migrate to the developing gonades. Okay. So primary oocytes have completed the first meiotic division by birth. That is also false. Primary oocytes are arrested in the prophase 1 of the meiosis at birth and do not complete meiosis 1 until just before ovulation. The majority of the primary oocytes become atretic by puberty. That is true. Around 6 to 7 billion oocytes are present in the fetal life, and they are reduced to about 3 lakhs to 4 lakhs at puberty, with most lost by atresia. Fewer than 10 follicles start the process of antrum formation in each ovarian cycle. That is false. Typically 10 to 20 follicles will begin to develop in each cycle, though usually one becomes dominant. The second polar body is formed at ovulation. That is false. The second polar body is formed only after fertilization, when meiosis 2 is completed. Okay. So oogenesis timeline: primordial germ cells originate in the yolk sac. Primary oocytes arrested in the prophase 1 from the fetal life. Meiosis 1 completes just before ovulation. Secondary oocytes arrested in metaphase 2. Second polar body is formed only after fertilization. So in fetal life, there are 6 to 7 million oocytes. 3 lakh to 4 lakh will be at puberty, and 400 to 500 ovulations in the lifetime. Okay. Next. Yeah.
Yes, sir. In the human testis, secondary spermatocytes contains 23 chromosomes. One
True or
secondary
false? Here options are not given. We will have
It is
to
a
take
true statement, sir.
true.
One
Okay.
secondary, yes, sir. One secondary spermatocytes form two spermatid. One secondary spermatocytes form two spermatid. No. No, it is a false statement, sir. The seminiferous tubules contain motile spermatozoa. No, no, sir. It is also false. The process of spermatogenesis takes 34 days. No, sir. It is also wrong. Inhibin is produced by primary spermatocytes. No, it is produced by Sertoli cells. It is also false. 1 second is
So
from four
one
spermatids.
one second one second primary spermatocyte will form two secondary, and two secondary will form further two. No, that is the way it should go. So one secondary spermatocyte.
One
Let us
spermatocyte.
see. Secondary spermatocyte contains 23 chromosomes. True. formed after meiosis, one primary oocytes haploid, one secondary spermatocyte forms two spermatocytes. Secondary spermatocyte, there will be two secondary spermatocytes, so total four spermatids will be formed. This is what secondary spermatocytes will give rise to meiosis 2, and two spermatids will be formed. Seminiferous tubules contain motile spermatozoa, that is false. Sperm living seminiferous tubules are immotile. Motility develops in epididymis. Spermatogenesis takes 34 days, that is false. Total duration is 64 days. Inhibin is produced by Sertoli cells. Okay, so secondary spermatocyte is haploid. Each secondary spermatocyte will give two spermatids. Motility acquired in seminiferous tubules is false. Spermatogenesis duration 64 days. Inhibin source is Sertoli cell. Next.
Yes, sir. In an X-linked pedigree, none of the sons of an affected male will be affected. None of the sons of the female will be affected. True. Half of the daughters of an affected male will not carry the gene. All will carry the gene. Half of the sons of the carrier females will be affected. Half of the sons of the carrier females will be carrier actually. Will be carrier, na? They will not be
Yes.
affected.
Yes.
Females are never affected. No, this is wrong. All of the daughters of a female carrier will themselves be carrier. No, half will be the carrier and half of them will be affected. No, so abhi kaise hoga? First, fifth one is wrong. Females are never affected. It is also wrong. Half of the sons of the carrier females will be affected.
Answer A.
Third, 3, 4, 5th is no. Half of the sons are also will not be affected. Half of the sons of the will be. They will
No,
be
half
carried.
of the females, yeah, half of the females will be, so that is also wrong, correct?
Wrong. Yes. First and second now. So none of the sons of an affected male will be affected. None of the yes. They will
Half
be
of the daughters on affected male will not carry the gene.
half of the daughters of an affected.
That is wrong.
Wrong.
All daughters will carry. Okay. So it should be A, no? A. None of the sons of affected male will be affected. Almost fourth time we are coming to that. The same question is twisted and asked in all different forms. Okay. So none of the sons of an affected male will be affected. That is true. Affected male will be sons will inherit Y from the father. Therefore, sons cannot inherit X-linked disorder from him. Half of the daughters of an affected male will not carry the gene. That is false. All daughters will inherit the father's affected X chromosome. 100% transfer that we have seen. Half of the sons of the carrier female will be affected. No, that is false. No, half of the sons of carrier females will be affected. That is true. Okay, but carrier half of the sons. Okay, that is true. Carrier 50% will be affected and 50% will be normal in cases of sons. Females are never affected. That is false. Females can be affected if uh, it is a homozygous affected or skewed X activation. All daughters of a female carrier will themselves be carrier. That is false. Carrier mother passes mutant X to 50% of the daughters. Okay, so X-linked recessive key rules: affected father 100% carrier to the daughter, 0% affectation of the son. Carrier mother 50% sons affected, 50% daughters carrier. Females can be affected, rare but possible. Okay, females never affected, that is false. All daughters of the affected male are carriers, that is true, and sons inherited disease from the father, that is false. Next.
Yes, sir. In the neonate, the appearance of external genitalia may not correspond with the genotype in the presence of androgenital system syndrome. Yes, true. Testicular feminization syndrome, true. Renal agenesis or Potter syndrome. No. So trisomy 21 syndrome. No, it is not there. Severe hypospadias. No, it is not there. Sir, option 1 and 2 is correct.
Severe hypospadias also they have given true. Okay, congenital adrenal hyperplasia, 46XX fetus, there will be virilization, male appearing external genitalia. So genotype is not same as phenotype. Testicular feminization syndrome, androgen insensitivity syndrome, 46XY, androgen resistance. So female external genitalia genotype will not correspond with the phenotype. Renal agenesis is a Potter syndrome, primary renal development disorder, not a disorder of sexual differentiation. So genotype and phenotype sex correlation is not affected. Trisomy 21 is false, chromosomal disorder, but no sex differentiation mismatch. Okay, severe hypospadias true, can be part of pseudo-vaginal perineoscrotal hypospadias, falls under ambiguous genitalia spectrum, may cause apparent mismatch between the external appearance and the expected male phenotype. Conditions causing genotype phenotype mismatch: congenital adrenal hyperplasia, androgen insensitivity syndrome, 5-alpha reductase deficiency, and severe hypospadias. Okay, DSD spectrum in okay. So CH XX virilized, AIS XY female phenotype, hypospadias may be part of DSD spectrum, and trisomy 21 no genital mismatch. Next.
Yes, sir. Chromosome analysis can be performed more quickly from CVS than from the amniotic fluid sample. Is based entirely on assessment of chromosome size. Is carried out at the metaphase stage of the mitosis. Of a classical hydatiform mole is usually 46XX. Of the fetus will be abnormal in about 80% of the cases of omphalos. Then we will move with CVS. Then no, the first one is wrong and is based on entire chromosome size. No, it is also wrong. It is carried out at the metaphase. This is true. Classical hydatiform mole is 46XX. It is also wrong. So first one is a.
So first is true, second is false, third one is true, fourth and fifth are false. Okay, can be performed more quickly. So CVS is faster than amniotic fluid. CVS allows earliest. Now whether it is a processing or whether it is a procedure, can be performed more quickly is different, and early gestation is different. Okay, so even this key is very dicey. Okay, CVS allows earlier diagnosis, that is one thing, 10 to 12 weeks, then amniocentesis, that is 15 to 18 weeks. Faster overall diagnostic turnaround. So overall you will get a quick report with
Yes.
CVS.
Okay,
But complexity of the procedure will be more with CVS. Okay.
yes.
So what is intended in the examination will depend upon how the question is framed. Okay, so don't go by the this only key, because sometimes the question's expectation may be different. Then based entirely on chromosome size, that is false. Karyotyping size uses size, banding pattern, and centromere position, not size alone. Okay, then metaphase stage, yes, that is true. Chromosomes are best visualized when condensed in metaphase. Interphase DNA has been copied before mitosis, but it still remains uncondensed inside one nucleus. Classical hydatiform mole is usually 46XX false. Complete mole is androgenic, paternal origin, not standard 46XX maternal normal diploid concept. Therefore, statement is false in this key system. Classical hydatiform mole is 46XX, no? This key, we need to
Yes, sir.
confirm
This
this
is
key.
the
This is a wrong key. This is the
wrong.
wrong key. Okay, this is a wrong key. Okay. Then exam follows fetal karyotype abnormal in 80%. That is false. Okay. Chromosomal abnormalities are seen in 50% of cases. Okay. Karyotyping basics done at metaphase, uses G banding, not based only on size. Complete mole is androgenic. No, this is wrong. Okay, partial mole is triploid. That is correct. Okay. Understand the concept. Don't assume that all keys are. Think about each and every key also. Next.
Yes, sir. In the neonate, the appearance of the external genitalia may not correspond with the genotype in the presence of. So we did just now. The repeated question 1 and 2 is 1, 2 and 5th is correct. Option A.
Option A. Okay. This is a repeat question. This is also repeat, I guess.
Yes.
This is also repeat. Here they have given the option. Yes, next 23.
Yes, sir. The following statements about inheritance are correct. Huntington's is a dominant gene. Phenylketonuria is a recessive gene. Hemophilia is an autosomal dominant condition. No. Von Willebrand's disease is a X-linked condition. No. Cystic fibrosis is an X-linked recessive. One is true. Second is true. Hemophilia is wrong. Third and fourth is wrong. So option A.
Option D, it should be, no? Option A, sorry. Option A. What is the second one? Second one I got.
Third one
Yeah,
is
correct. Option, yeah. Yes. So, Huntington's chorea, autosomal dominant, phenylketonuria, autosomal recessive, hemophilia, X-linked recessive, von Willebrand's, autosomal dominant, cystic fibrosis, X-linked recessive, that is false. Okay. And CFTR gene, chromosome 7 is involved. Okay. Next, question 24.
Concerning a woman with blood group type A, she may have a genotype AB. Her father, she may have a genotype of AB. Her father may be of group O. No. All of her children will be group A or O. If her husband has a blood group A, all of her children will have a group A or O. She can be transfused safely with blood group AB. Okay. Fifth one is wrong. AB is a universal receiver, not the donor. So fifth one is wrong. She may have a genotype AB. AO. She may have a genotype of AO. So first one is wrong. Her father may be a group of O. A or O. It is true.
Yes. Yes.
All of her children will be group A or O. Will be A or all of her children will be group A or O. It can be it will be depending on the paternal wala.
Yes.
Or not. So this may be true or false.
That cannot be commented. So, okay.
If her husband has a blood group A, all of her children will have a blood group A or O. If he also has, yes, this is true.
Yes.
If
yes.
both having A, then
Here
A
they
or
have given in here they have given husband's blood group. So this we can say, but here we cannot comment anything. Okay. So that pattern is FTFTF. Okay. So she may have a genotype AB, that is false, blood group A. So either it will be A or AO. Okay. So the statement is false. Her father may be group O, that is true. Father's genotype may be OO. He can pass O allele only. Okay, daughter still can be group A if the mother contributes to the A allele. Okay, therefore possible. All children will be group A or O, false. Depends on the father's genotype. Okay, if father is B or AB, then children may be B or AB. Okay, so all children statement is incorrect. If the husband has blood group A, all children will be A or O, that is true. Okay, then she can be transfused safely with AB, that is false. Okay, so blood group A genotype will be AA or AO. Blood group B genotype will be BB or BO. AB will have AB only and O will have OO genotype. Okay, O is universal donor, AB is universal recipient. A receives A or O only, A has got a anti-B antibodies. Next.
Concerning chromosomal errors, structural variations may not have a phenotypic structural variations may not have a phenotypic consequences. Trisomy indicates the presence of 3 haploid components within the cell. Yes. Non-disjunction during mitosis can contribute to mosaicism. No. During meiosis, it contributes to mosaicism. A trisomic parent can produce normal gamete. A trisomic patient can produce a normal gamete. No. A triploidy is rarely detected in neonates at term. Triploidy is rare. This is also wrong. So first, sexual variation may not have a phenotypic consequences. It can have phenotypic consequences. Option 1 is wrong. Trisomy is true. Second is true. Third one is false.
No, the keys is a true, false, and all are so except second, rest all are true. Okay. So trisomy indicates the presence of 3 haploid components within a cell. Let us see the key. Structural variations may not have phenotypic consequences. That is true. Reciprocal translocations often involve no net loss of genetic material. So there can be balanced rearrangements. So phenotypically it will be normal. Okay. So structural variations may not have phenotypic consequences. That can be true. Okay. Then trisomy, 3 haploid components, that is false. Trisomy will have 2n plus 1 extra chromosome, so not 3 haploid sites. Okay, that is 3 triploidy will be equal to 3n. Non-disjunction in mitosis causes mosaicism. That is true. DNA has been copied before mitosis, but it still remains uncondensed inside the nucleus. So post-zygotic mitotic error, there will be 2 cell lines, leads to mosaicism. Trisomic parent can produce normal gametes. That is true. Meiosis in trisomy can produce normal gametes or aneuploid gametes, therefore true. Now here this little trisomy parents can produce normal gametes. Meiosis in trisomy can produce normal gametes and aneuploid gametes. So there will be in meiosis there will be normal gamete and aneuploid gamete. So normal gamete may be produced. Okay, so some normal gametes may be there. Then triploidy rarely detected in the neonates. That is true. Most triploidy causes early miscarriages or stillbirth, and survival is rare. That is why it is true. Okay. So this is what we have seen. Next.
The following conditions are hereditary. Polyposis coli, yes, retinoblastoma, autosomal dominant, xeroderma pigmentosa is no, Burkitt's lymphoma, osteosarcoma, HNPC coli. Retinoblastoma is true. HNPCC and xeroderma is actually a mutation based on and 3 hereditary. Third option C.
We'll see the option. Your answer was C. The option given is A. Answer given is A. Last 2 are false. Burkitt's lymphoma and osteosarcoma. Okay. Polyposis coli is true. Familial adenomatous polyposis, autosomal dominant. Retinoblastoma is also true, autosomal dominant. Xeroderma pigmentosa, yes, true. Then that is autosomal recessive. Burkitt's lymphoma, sporadic or infection associated with Epstein-Barr virus. Okay, and osteosarcoma is false. Mostly sporadic, may be associated with RB1 or TP53 mutation, but not necessarily hereditary disease itself.
Yes, sir. The following are autosomal recessive diseases: neurofibromatosis, cystic fibrosis, phenylketonuria, Polyposis coli, sickle cell anemia. 2, 3, and 5th is there. HNPCC is autosomal dominant. So fibromatosis remains. 2, 3, and 5th is true. What option A, sir?
Option A. Neurofibromatosis is false. Autosomal dominant. Cystic fibrosis is recessive. Phenylketonuria is recessive. Polyposis coli is autosomal dominant. Sickle cell anemia is autosomal recessive. Okay. Next.
The primary oogonia undergoes.
Yeah.
The primary oogonia undergoes mitotic divisions prior to meiosis. Primary oogonia undergoes mitotic prior to meiosis. The primary oocyte enters meiosis at puberty. At birth, primary oocytes have completed the first meiotic division. The DNA synthesis does not take place as a part of the second meiotic division. Secondary oocytes complete the second meiotic division at ovulation. 5th one is true, sir. And the primary oogonia undergoes mitotic prior to the meiosis. First one is also true. The primary enters the meiosis at birth. No, the second one is false. We have A and B. The at birth primary oocytes have completed the first. It is also false. The DNA synthesis does not take place as a part of a second meiotic division. It undergoes directly meiotic division. So it is true. We will complete the second meiotic. True.
The last one is a
Secondary oocyte completes the second meiotic ovulation.
that is
Yes.
false. That is false. No, at fertilization,
Yes,
fertilization will happen. No. So primary oogonia undergo mitotic divisions prior to meiosis. That is true. Oogonia proliferate by mitosis in fetal life. Okay, but this will happen only in the fetal life. Again, this is little controversial. Okay, so they enter meiosis 1 to become primary oocytes. Primary oocytes enter the meiosis at puberty. That is false. Meiosis begins in the fetal life. Arrest occurs in prophase 1 until puberty. At birth, primary oocytes have completed first meiotic division. That is false. At birth, primary oocytes are still in the prophase 1 arrest. Meiosis 1 is not yet completed. DNA synthesis does not take place in second meiotic division. That is true. Okay. Secondary oocytes complete second meiotic division at ovulation. That is wrong. Secondary oocytes are arrested in metaphase 2, completes meiosis 2 only after fertilization. Next.
Yes, sir. Primary spermatocytes undergoes reduction division. True. Each primary spermatocyte ultimately gives rise to 4 spermatid. This is true. The whole process of spermatogenesis in man takes 6 to 7 days. The whole process of spermatogenesis. No. The grossly abnormal spermatozoa may found in fertile grossly abnormal sperm may found in fertile semen. Spermatids are haploid. This is true. Undergoes the primary. First one is true. Second one is true. Third one is false. May be found in grossly abnormal spermatozoa. May be found. Maybe. It is also true. Maybe it can be found.
Yes. So option A. Okay. So primary spermatocytes undergo reduction division. That is true. Interphase DNA copies once before meiosis, while chromatin stays uncondensed. Primary spermatocyte undergoes meiosis 1 reduction division, produces secondary spermatocytes, that is haploid. Each primary spermatocyte gives rise to 4 spermatids. That is true. One primary spermatocyte will give rise to 2 secondary spermatocytes. Okay. And each will give rise to further 2 spermatocytes. That is why from one primary spermatocyte you will get 4 spermatids. Remember what you have been asked, from here to here or from here to here. Okay. Then spermatogenesis takes 6 to, that is false. Grossly abnormal sperm may be found. Yes. Normal semen contain abnormal forms also. That is also to certain extent is normal. Variable morphology. Fertility depends on overall functional sperm count, not perfection. Spermatids are haploid. True. Okay. Next.
Yes, sir.
Read it.
The following Yes, sir. The following are structural aberration of chromosomes: deletion, inversion, aneuploidy, polyploidy, translocation. 1, 2, and 5th is true. So option A, sir.
Deletion is a chromosomal or what exactly? Structural? Yes, structural aberration of chromosome. Okay,
Division. Option A.
structural aberration of chromosome.
Option A, sir. Deletion is there, translocation is there, and inversion.
And inversion is there. Okay. Otherwise, aneuploidy and polyploidy, they are with respect to numbers. Okay.
Number.
Okay. So third and fourth are false. Rest are all true. Deletions, yes. Loss of chromosomal segment, Cry-Due-Chart syndrome, 5p deletion. Okay, chromosomal segment reverses within the same inversion. Chromosomal segment reverses within the same chromosome. No net gain or loss of genetic material. Aneuploidy is false. DNA copies once before meiosis while chromatin stays, and this is something different. Polyploidy is false. Whole extra sets of chromosome, that is 3 and 4 and etc. Also numerical abnormality, not structural. Translocations is true. Segment exchange between the two non-homologous chromosomes. Okay, can be balanced or unbalanced. So structural abnormality will be deletions, inversions, and translocations, and numerical will be aneuploidy and polyploidy. Next.
Messenger RNA synthesis is dependent on RNA polymerase, is an exact copy of sense DNA, contains exons, is measured by Western analysis. Translation occurs in the nucleus. Synthesis, it is true. Exact copy of sense DNA. Not the exact copy actually. Contains exons. 1 and 3 is true. It's measured by Western analysis. It is false. Fourth one is false. Translation occurs in the nucleus. This is but it is transcription. Messenger RNA is associated with transcription. So it is also false. Fourth, fifth is false.
So the correct one are the first and third is only correct. So first is correct, third is correct. Okay. So contains exon and synthesis dependent on RNA polymerase. These are the only two correct. So synthesis depends on RNA polymerase, that is true. Okay, template DNA will lead to the mRNA. Okay, then exact copy of sense DNA, it is false. mRNA is complementary to template strand. It is a complementary copy, not the same copy. Okay, then if DNA, if it matches coding strand, not sense DNA as exact DNA copy, contains exons, true. After splicing, introns are removed and exons retain in the mature mRNA. Okay, measured by Western blot, that is false. Western blot is for proteins. mRNA is measured by Northern blot or RT-PCR. Okay, translation occurs in nucleus, false. Translation occurs in the cytoplasm. So transcription happens in the nucleus, mRNA processing is in the nucleus, and translation is in the cytoplasm. Okay, Western blot is for proteins, Northern blot is for RNA. Next.
Yes, sir. A woman has a rhesus genotype of CDE /CDE, can develop anti-C antibodies. Okay, could safely be transfused with D positive blood. Okay, may develop anti-E antibodies. Should receive anti-D immunoglobulins after giving birth to RHD should receive anti-D immunoglobulin after giving RHD positive infant. Anti-D immuno after giving birth to RHD positive infant is RH negative.
Is she RH negative? This lady?
No, she is RH positive. No.
She is RH positive. So this is wrong. Okay. Should receive anti-D immunoglobulin? No. Okay. May develop anti-E antibodies. She can develop anti-E antibodies. No. Okay. Could safely be transfused with D positive blood? Yes, she can be transfused with D positive blood. Could develop anti-C bodies? She cannot develop anti-C antibodies. Okay, CC is there. Okay, so that is false. Okay, could develop anti-C antibodies, that is false. She develops, she already carries C antigen. Okay, you do not form antibody against self antigen, so anti-C cannot be formed. Could safely be transfused with D positive, true. She is Rh positive. Okay, may develop anti antibodies, true. DNA, she carries E antigen. Okay, but immune system can still form allo antibodies after exposure. Okay, then should receive anti-D immunoglobulin, no. Is Rh negative, false. She is Rh positive. Next.
Is diseases determined by dominant gene? A sibling of an affected person will always be affected. A sibling of an affected person
Dominant
will
gene, now we are talking. Yes, sibling of affected person will always be affected. Homozygous affected parents will always produce affected offspring.
of
Well,
hmm.
will always
the
produce.
parent,
That is also true.
yes,
Yeah.
true. Parents of affected person have an increased rate of consanguineous marriage. True, it
Not
can happen.
necessarily, no.
Not necessarily.
It
It
is
can
an we are
happen.
dealing. Yeah, it can happen. Even if it is a non-consanguineous marriage, then also
So it can
the
happen.
okay.
Normal children of affected parents have normal offspring. Normal children of affected parents have normal offspring. New cases can arise. It can. Fourth one is wrong, sir. Even if the children is normal, they can have affected children. New
Normal
cases can
children of affected
affected.
parents have normal offspring. That should be true, no? If it is a
That
dominant,
hmm,
if it is a dominant, there is a normal children. That means it is not there, no? It is a dominant
there.
disease. So their children will always be normal. So it will be true. New
New
cases
cases
can arise by spontaneous. That is also true. Okay.
true. Hmm. Yes.
So answer key is what is false? Yes, the parents of affected, last two are true. The parents of affected persons have increased rate of, not necessarily, that we have seen. Okay, this is false, and first is also false. A sibling of an affected person will always be affected. A sibling of affected person need not, because it is a dominant gene, no, even it will be a single gene will be affecting. Not all children will be affected, no, 50% will be affected. Sibling, there will be 50% risk. Heterozygous affected parent and normal parent, there will be 50% risk. Okay, sibling of affected person will always be affected, no. Okay, it is an autosomal disease. There will be 50% chance of affection of the child. Okay,
Hmm.
hello doctor, you getting?
Yes,
No.
sir. Yes, sir.
Okay, homozygous affected parents will always produce affected offspring. Yes. Okay, true. Increase consanguinity, not necessarily. Normal children of affected parents have normal offspring. Yes. Okay, new cases can arise by spontaneous mutation. That is also true. Okay, next question.
In the human haploid cells, cells contain homologous pair of chromosomes. Polyploid cells contain an extra set of chromosomes. Monosomic cells contain an extra chromosome. Haploid cells contain 22 autosomes. Chromosomes divide during the S phase of the cycle. Haploid cells contain homologous pairs of chromosomes. No, this is wrong. First one is false. Polyploid cells contains an this is true, extra set of chromosomes. Yes, this is true. Monosomic cells contain an extra chromosome. This is false. Haploid cells contain 22 autosomes. This is true. Fifth one is false.
So first false, third false, and fifth false. So second and fourth is only true. Okay, second and fourth is true. Polyploid cell contains an extra set of chromosomes, and haploid cells contains 22 autosomes. These are the true sentences. Okay, haploid cells contain homologous pairs. Haploid cells have single set of chromosomes, no homologous pairs. Okay, then polyploid cell contain an extra set of chromosomes. True. Okay, monosomic cells contain an extra chromosome. False. Monosomy, there is a loss of one chromosome. Haploid cells contain 22 autosomes, that is true. Chromosomes divide during S phase, that is false. S phase is a DNA replication. Chromosome separation occurs in the mitosis, M phase. Okay. Haploid is n equal to 23 chromosomes. Diploid 2n is 46 chromosomes. Monosome is 2n minus 1. Trisomy is 2n plus 1. Polyploidy is 3n or more. S phase is a DNA replication. Next.
35 chromosomes are found in the same number in all mammalian cells. Can be analyzed more quickly from a blood sample than from the amniotic fluid sample. Can be reliably identified by the lens. The Y chromosome is larger than the X chromosome. DNA content is doubled during the S phase of the cycle. Fifth one is true. The Y chromosome is larger. No, it is false. Can be reliably identified by the length. This is true, sir. Can be analyzed more quickly from the blood sample than from the amniotic fluid sample. This is also true. Are found in the same number in all mammalian cells. No, in oocytes and they are in the haploid number. Oocytes and spermatocytes. Option A, sir. Option B.
Option A. Option A, same number in all mammalian cells? No. Somatic cells are diploid, 46 chromosomes in humans. Genetic gametes are haploid. Okay, so chromosome number is not the same in all cells. Faster analysis from blood than amniotic fluid, that is true. Identified reliably by length, that is false. Then Y chromosome larger than X, that is also false. X chromosome is larger, Y chromosome is smaller and gene poor. DNA content is double during S phase, that is true. So S phase DNA replication happens. Okay. Next.
Cell cycle or DNA synthesis. The principal phase of DNA synthesis is G1. A tetraploid quantity of DNA is present at the end of G2. G2 is the post-mitotic resting phase. The cells are generally sensitive to anti-metabolite in the S phase. The DNA is completely replicated several times. Fifth one is wrong. It's completely replicated several times. Fifth one is wrong. The principal phase of the DNA synthesis is G1. No, first one is wrong. A tetraploid quantity of DNA is present at the end of the G2. Doubles the chromosome. Option 2 is correct, sir. G2 is the post-mitotic resting phase. No. Third one is false. Cells are generally sensitive to antibiotic. This is true. Complete. Option A, sir.
Option A. Okay, so second and fourth is correct. DNA synthesis is mainly in the G1 phase, that is false. DNA synthesis occurs in S phase. Okay, then tetraploid DNA at the end of G2, that is true. DNA content doubles in S phase. After S phase, 2n equal to 4n DNA content. G2 therefore contains tetraploid DNA content. G2 is post-mitotic resting phase, that is false. G2 is a pre-mitotic growth phase. Cells sensitive to anti-metabolites in S phase, true. Okay, DNA is completely replicated several times, that is false. DNA replication occurs once per cycle, prevented by cell cycle control mechanism. So G1 is a growth phase. S phase DNA replication happens. G2 pre-mitosis preparation. M is a cell division. G0 is a resting phase. Next.
Yes, sir. The following are structural abnormalities of chromosomes: deletions, duplications. Sir, this is a repeated one.
Repeated question.
Yes.
Yes, it is a repeat question. Okay.
Yes.
Okay, that finishes the topic of genetics. Thank you.